did MSc from Bangkok University of Thailand, and she got BSc from Islamic University, Malaysia. She has specialization in halal sciences, food products, biotechnology, molecular biology, and tissue culture.
Yes, Dr. Najwa, over to you, please.
Dr. Najwa, are you
Yes, can I
listening?
start now?
Yes, yes, you proceed,
Okay.
please.
Assalamualaikum warahmatullahi wabarakatuh, and good afternoon to all of you. Thank you for inviting the Halal Science Center Chulalongkorn University to sharing our experience with you. So today I will be talking about analytical halal methods for determination of fatty acid and ethanol halal traceability of Thailand's experience. So nowadays, raw material used for producing halal food presented in many forms. So halal food product could be adulterated with haram ingredient at any point in the food chains. So there are many complexity of novel sample, as you can see in here. That some of example like there is protein bars from the insects and plant-based meat, as well as the edible growth, which is coming from 3D printings. So nowadays, the food become more complicated by the new technology, which beyond the perception of the consumer. So the adulteration of non-halal ingredient could not be seen by the naked eye. This is where the halal forensic science laboratory play a crucial role in supporting the Islamic organizations. So for Thailand, we have a strategy to be kitchen to the world. So we are very concerned about halal foods as a kitchen in the world. So Muslim as a minority, which are living as a minority, we are highly aware of adulteration of haram ingredient in the food products. So during 1994, there is a rumors in Thailand about using lard oil for coating Asian rice noodle, was spread over Thai communities. So at that time, Dr. Vinay Dahlan, as a scientist, start to exploit the knowledge of science and technology to answer the issue by utilizing the facility provided in Faculty of Allied Health Science. And then after analyzing, we found out that the fatty acid profile of the noodle containing oil by gas chromatography, it revealed that it's not contain any lard oil. So this is the first of instrument we are using as the halal in the halal science center to detect the non-haram ingredients. So this is how scientific is very useful in answering the Islamic procedure and principles. So for the gas chromatography, is one of the most popular method for separating and analyzing compounds. So this chromatographic technique where the mobile phase is gas, in here usually we use helium gas, which required analyte to be either natural volatile or can be converted to a volatile derivative. So the advantages of this instrument is high resolution, and it has a very speed and high accuracy, and it can be reproducibilities for many samples, and moreover it also ease of preparation and operations. And for the gas chromatography overview, the TC or gas chromatography system consists of gases with pressure and flow regulator, sample injection system, chromatographic column, detector, and computer or recorder. At first, sample will be injected through the septum into heated injection port, and then the sample will be rapidly evaporated and is pulled through the column via the carrier gas, and the column will be heated to provide sufficient vapor pressure to elute the sample mixture, and then the sample will be mixed flow through the heat detector for determinations. Oh, sorry. And this is an example of chromatogram of standard uh, of a mixed sample comparing with the unknown sample. So let's say if you have the sample containing the same compound with the standard, it will show that the same retention time as the standard, like an example of compound A and B in the figure. So the retention time is a measure of the time taken for a solute to pass through a chromatography column. So it can be calculated as the time from the injection to detections. And this is an example of a standard frame of fatty acid methyl ester comparing with the palm oil example. And this is an overview of method using for fatty acid analysis and alcohol analysis. So if you can see from the overview, it can say that both fatty acid analysis and alcohol analysis can be used by can be interpreted by gas chromatography. So for the fatty acid, it separated by gas chromatography, which are different in fatty acid profile depend on the source of origins. So at first, the sample for fatty acid determination will be extracted by solvent extraction, and then it can be catalyzed by acetyl chloride or boron trifluoride to be a derivatized sample, and then it will be separated by carrier gas helium, and later on we can interpret from the data from the chromatogram given by the computer. And for the alcohol, usually it needs that the sample should be homogenized and then filtrated before added the internal standard. And then we also have to prepare the ethanol standard for quantifications. And then we separated the sample by gas chromatography by the same principle as the previously mentioned. And then later on, we will be interpreted data by comparing the calibration curve with the sample given. For the analysis of fatty acid, the scope of this analysis is usually we are sampling the food products such as meat, vegetable oil, milk, and chocolate or other products. And then the principle of this is at first, we have to undergo the methylation or derivatization to be fatty acid methyl ester, and then separated by gas chromatography. So it is very important during the sampling of the sample. We have to make sure that sample is homogenized and make it reduce the particle size and maximize the surface area. So we have to ground the sample, and then we have to add dichloromethane methanol in the ratio of 2/1. And then after we added the dichloromethane and methanol, we have to stand for 1 hour. Usually we have to vortex every 15 minutes to make sure that all samples mix well. And then later on, after 1 hour, we have to filtrate the sample with Whatman #1 paper. And then we have to add the solution KCl 0.1 molar of KCl and centrifuge at 2000 RPM to separate the pure lipids. And later on, we have to discard the supernatant. And then we take the sample from the previous one. We use that sample for derivatization. So this sample will be transferred to the new test tube, and the sample 200 microliter of the sample or 220 microliter of the oil will be added to the new test tube, and then the new solvent will be added. Uh, um, Methanol and hexane 4/1 will be added, and the catalyst acetyl chloride will be added to the sample. And then we have to boiling at 100 Celsius for 1 hour. And later on, we will have to centrifuge, we have to add potassium carbonate, and then we have to centrifuge at 1000 RPM. And after we centrifuge, we can see the fraction, two fraction easily as shown in the slide. And then this true fraction will be, we only take the supernatant to be injected in the GC. We added in the vial and put in the GC gas chromatography. And this is how to interpret the result. So we have to calculate the fatty acid percentage by its area under the curve, and divided by total area of all fatty acid sample and multiply by 100. So we get each fatty acid content of each percentage of each sample. Sorry, actually I have video, but I will show you later. And next, for the alcohol. So this is the example of this is an example which of the food product that containing alcohol, especially the ethanol. So you can see that alcohol can be found in the flavors as the solvent, and also it can be found in some beverage as a solvent as well. But the other alcohol that could be found is in the fermented food, like vinegar, or in soy sauce, soya sauce. But the alcohol that should not be used is alcohol which is coming from wine. So according to our regulation in Thailand, alcohol processing for halal food production should not coming from fermentation as such as wine, beer, or beverage containing liquid that were not permissible. And then the alcohol used as a solvent is permitted uh, unless that the final concentration of the alcohol in the food product or beverage must be less than 0.5%. And for the natural products such as fermented food, it's permissible if alcohol containing products derived from the fermentation is less than 1%. And for the alcohol analysis, usually the food or beverage used for this analysis is soy sauce, energy drink, fruit juice, vinegar, and artificial flavors. So the principle of alcohol analysis is ethyl alcohol in the drink will be diluted by 0.02% volume by volume of N-propanol as an internal standard. And then it will be analyzed by gas chromatography by flame ionization detector, and the amount of alcohol in the sample would be compared with the calibration curve. So at first, before you start anything, of course, when you have to do laboratory things, you have to prepare the reagent and also standard reagent before starting any laboratory works. And then later on, you have to prepare the sample, make sure that the sample is ready for the testing, and then testing the sample, and later on will be computation and analyze the sample by computer. So for the reagent, here we prepare the internal standard of N-propanol and also standard of ethanol with 8 concentration as shown in the slide. So for more detail, usually we add 1 ml of the sample. Usually we added 1 ml of the sample, and then 1 ml of 0.02% of N-propanol in 20 ml tube, and then mix together. But you have to make sure that whenever you do alcoholic analysis, you have to make sure that the sample doesn't have any particle inside. Otherwise, you have to filtrate the sample before transferring the sample and add it with the internal standard. And then when the sample is really added, it can be directly after filtration with 0.445 micron of filter, then we can directly analyze the sample by gas chromatography. And this is how to quantitative analysis of the alcohol sample. So we can compare the sample with the calibration curve that we are doing. As you can see from here, that the example of ethanol and unknown sample. You can use from the graph to calculate the concentration of the sample. And later on, you want to know how many percentage of the ethanol in the sample weight by volume. So you have to calculate it. After you get the concentration of ethanol from the calibration curve, you have to multiply the sample with 0.789. This is the density of the ethanol. Okay, this is the other video, but I tried to open it, but it could not be work very well. So I will try to open outside the presentation. And this is our published books and papers. So if you are curious about how we are doing our routine works here, you can go through this paper. It has many detail in how we extract the sample and how we prepare the sample. So let me open the video. So I just want to show the overview how we prepare the sample. So this is for fatty acid analysis. So this is a gas chromatography. Huh? Okay, wait. Yeah. Okay, sorry, just now I forgot to share the video. Okay, this is our laboratory. So I will show you our gas chromatography machines. So she's explaining about how we prepare the fatty acid profiles. As I told you just now that we have to grind the sample and make it homogenized. And then added extract solutions in the sample. We have this machine to incubate the sample for 1 hour. And then we filtrate the sample to get the liquid solutions. And later on, yeah, we do derivatization by adding methanol and hexane and acetyl chloride. You have to be careful when using acetyl chloride. Yeah, and then we transfer the sample in the vial and inject into the gas chromatography. This is another video for preparing of alcohol. So she added the sample mixed with the internal standard. And later on, after mixing, she will be filtered with the 0.45 micron filter. Yeah, and added to gas chromatography. So it's not very difficult actually for the alcohol analysis. So as long as you have gas chromatography, that's all from me. Thank you.
Hello, Dr. Najwa, I have completed.
Yes. I'm done. Thank you.
Thank you. You presented very nice deliberation. Now the session is open for questioning. Do you have any question?
Assalamu alaikum ji. It was really nice presentation. My name is Dr. Imran Khan, working in Pakistan Standard Quality Control Authority. Is there any conventional method for fatty acid isolation, separations, other than gas chromatography?
Pardon, can you repeat your question?
My question is there any conventional method as that now you are using gas chromatography method? So is there any easy and conventional method that we can easily use in lab for the separation of fatty acids? I think.
There is no commercial method, yeah, for this. But actually, you can use if you cannot use for alcohol analysis, you can use HPLC also, yeah, instead of gas chromatography. Yeah.
Additionally,
Or
you
maybe
can
you can use TLC, thin layer chromatography for simple interpretation of the if your sample is not too complex. Yeah.
Dr. Najwa, can you hear me? Am I audible? Can you hear me?
Yes.
Okay.
Can you hear me?
So, question, because Dr. Vinay Lab is a very well-known lab for the analysis of forbidden substances and in halal research. So, as far as the lard oil concern, I mean, do you have a kind of method or practicing a method by which you can differentiate the mixing of lard with tallow or with other animal oil? I mean, do you have a method by which you can differentiate it or differentiate mixing of two animal oils if it is mixed with lard or something like this?
Oh, okay. Actually, in our papers that I show you just now, it explaining about C16 and C18 one. We use that ratio to is actually is our own experience that we have analyzed that C16 over C ratio of the C16 and C18 over 1 should be less than if less than 0.6, it will be similar to the last sample. But if it more than 0.6%, it will not be contaminated with last sample. That's what our experience that we have done in our laboratory. If you want to see more detail, I think better you go to our papers. We have discussed about the how we interpret the fatty acid profile from that. Yeah.
Thank you.
Thank you, Dr. Najwa. Have a nice day. Thank you very much. The next speaker is Dr. Madiha Isra. She's from Turkey. Dr. Madiha Isra is a molecular biologist and has specialization in genetics. She graduated from Middle East Technical University of Art and Science, Department of Molecular Biology and Genetics in 2008. She received her PhD in biochemistry from University of Houston, USA in 2012. She has been working in Tubetic, Marmara Research Center Food Institute as a chief scientist since February 2013. She is the head of Food Molecular Biology Laboratory. She has been working on DNA and protein-based detection system, DNA isolation methods, generation of monoclonal antibodies, development of epitomers, development of lateral flow first step, and rapid sensory system. Now I request Dr. Isra to proceed. Our topic is rapid on-site halal detection methods. Dr. Isra, please.
Thank you. Am I audible?
Yes, yes.
Okay, thank you. Assalamu alaikum wa rahmatullahi wa barakatuh to all. Dear distinguished participants and dear sisters and brothers, it's a pleasure for me to be here today. So thank you for all coming here and listening our courses. So today I will talk about rapid and on-site halal detection systems. Basically, we know that for halal analysis in laboratories, we identify non-halal ingredients by majorly PCR, GCMS, LCMS, HPLC, and FDIR methods, and there are more methods too. They are very reliable and very sensitive methods, but the problem with them is the complexity of the experiments and requirement of experienced technical personnel and well-equipped laboratories also. And each method, each analysis is highly costly. And that's why we think that with the recent scientific advancements, we say there is another way. So in the modern world, we see like robots and automated systems all around the world in all areas. So we believe that it is time to establish a halal control analysis outside the laboratories in miniaturized systems so that it will be available to everyone. So why it is important to use biosensors in halal food control? Halal certification is a long and sensitive process. It covers the product and the production process. So rapid and on-site control of each and every component will facilitate the process and maintain the safety and quality of the product. So it is better to have a real-time monitoring. So from another point of view, from the consumer side, in the globalized world, Muslim consumers might be able to check the halal status of the food they eat everywhere. So when they are in the restaurants, on planes, and at home, so they will be able to check their food, and they will be their own auditors. And in another point of view, in Islamic countries, halal status of every product will be able to be checked rapidly on customs. So there will be no suspicion, no doubt about the materials imported through customs to Islamic countries. That means we're going to talk about biosensors today. So the IUPAC definition says that biosensors is a tool that uses specific biochemical reactions to detect the chemical elements of a substance, usually electrically, optically, or thermally, through isolated enzymes, tissues, or cells, or I can add here through antibodies, aptamers, some polymers, and so on. So actually the concept of biosensor was first developed by Dr. Leland Clark in early 1960s to measure glucose concentration in diabetic patients by glucose oxidase enzyme. It is probably the most common biosensor all around the world. Probably some of us have those kind of biosensors at home. So actually today I will try to give you a brief information about biosensors that are used in halal food control. And I will try to tell you some aspects on how we should design the future of halal food control as the researchers here. So let's start with alcohol biosensors. So alcohol biosensors are fabricated with either enzymes or bacteria. As the bacteria, they use Acetobacter species, but most of the alcohol biosensors uses enzymes though, either alcohol dehydrogenases or alcohol oxidases. And both of the enzymes operates by cofactors, and there is an electron transfer which can be either visually seen by color change or red, the color change is red measured, or the electron transfer is measured as a signal by electrical devices, electrochemical devices. So when the amount of alcohol increases, they see increases in the signal too. Here you can see the increase. And as a simple example of an alcohol biosensor, they used alcohol oxidase immobilized on polyaniline. The sensitivity of this biosensor is down to 1% ethanol, which they say that it is in line with the Islamic dietary law because the food itself also contains some sort of ethanol because of the fermentation processes. So it's a very simple stick actually. You just dip it into the sample solution and visually wait for the color change. So for the rest of the presentation, I will try to talk about biosensors for non-halal meats and its derivatives. So let's start with gold nanoparticle biosensors. They can differentiate the pork from beef and poultry by visual color change. Normally, gold nanoparticles in non-aggregated form, they are in red in color. But when they are aggregated, they become a purplish, bluish purple color, let's say. And when there is a microDNA present in the sample with the gold nanoparticle, this microDNA is very specific for porcine DNA, and it specifically detects the porcine DNA's presence. So this is a single-stranded microDNA, and it goes and binds around the gold nanoparticles and keeps them away from aggregation. But when after the addition of the porcine DNA as the sample, this microDNA goes and binds to the porcine DNA specifically, and it becomes the double-stranded DNA, leaving the gold nanoparticles naked. In this form, with the addition of the salt solution, gold nanoparticles start aggregating, and they form the color of this bluish purple. This is a very basic biosensor though. So normally you have the gold nanoparticles with the marker added inside. You just put your sample in and add the salt solution and wait for the color change. This sensitivity of those types of biosensors are maximum 1%, but there are some studies with the sensitivity around like 20% porcine mixture. So it's not very sensitive though, but like if you're a consumer then buy some meat from the butcher and want to check it, so it's a good and basic way of a biosensor. The problem with this biosensor is that it requires DNA isolation as the preliminary preparation step. So let's see another example of gold nanoparticle biosensors. So in this study, they used porcine marker with a fluorescent dye tagged on it. And when it is when it is bound around the gold nanoparticle, the gold nanoparticle quenches the fluorescent dye. When the target DNA, the porcine DNA, is added, it binds to the marker DNA, taking the fluorescent away from the gold nanoparticle, and then the fluorescent dye starts to give the fluorescent signal. And when there is an increase in the sample, the signal increases. So as you can see here in the plot, when there's 100% pork inside the sample, the signal is really high. When they decrease it down to 1% pork, they still have the detection. And when they have other animals' meat, it's just the baseline and they don't have a detection. So let's move to lateral flow test strips as another way of biosensors. They can verify the presence of pork by pork-specific antibodies. And if those antibodies are specific to raw or processed food, so this test strips can detect either raw or processed foods too. They're very easy and very simple to use, and they have the result within like 10, 20, maximum 20 minutes. A very basic example of lateral flow test strips are pregnancy tests. You know, it's very easy to use. You just put the sample on it and wait for the result. If there are 2 lines, that means it's positive. If there's 1 line, that means it's negative. And if you don't see any line, that means it's a failure. So you have to repeat the tests with another test strip. So how does it work? You can see the V-shaped antibodies here attached to the gold nanoparticles. These antibodies are very specific to pork, and the same or another pork-specific antibodies are also immobilized on the test line, on the test strip. And there is a control or comparison line. These antibodies are secondary antibodies that can bind to the antibodies that are attached to the gold nanoparticles. So the concept of this lateral flow test strip is the basic paper chromatography. When the sample is added, the porcine protein inside the sample is caught by the porcine-specific antibodies here. With the flow of the liquid, they start moving towards the test line. When they reach the test line, if there is porcine protein, the test line antibodies catches the porcine proteins and making kind of a sandwich here. With the presence of the gold nanoparticles here, we see a red color. And some of the remaining antibodies that are not attached to the porcine proteins are caught by the control line antibodies, the secondary antibodies. So they also form the second red line here because of the presence of gold nanoparticles again. So if you see 2 lines, as in the case here, that means the sample has porcine. We actually had a study of lateral flow test strips. We developed an antibody against one of the porcine proteins. And we try to see how it works in the lateral flow test strips. So since it was just a laboratory test, we don't have the control line here. It's just a test line. We had a little bit of porcine meat, and we dissolved it in PBS buffer, and we wait for the liquid to go through the test strip, and we saw the presence of the red line here. Let's move to the electrochemical biosensors. They're actually, I believe they're going to be very common in next years all around the world in all areas probably. They're portable, very easy to use, and cost-effective equipments. They are highly specific and sensitive. Their sensitivity levels are generally low. They can detect lower than ppb levels, sometimes like picomolars, femtomolars, and etc., depending on the sample type. They have a very minimal sample preparation. You just like dissolve the sample and put it on. They are miniaturized analytical devices. There are actually like a few types of electrochemical biosensors, such as like blood glucose sensors are unparametric electrochemical biosensors. Like pH meters are like potentiometric ones, and there are some conductometric biosensors, piezoelectronic ones. And today I will not be able to talk about all of them, but I will try to give you some ideas. So electrochemical biosensors converts a biological response into an electrical signal. So the biorecognition molecule is immobilized on the electrode. When the analyte comes and binds to the recognition molecule, they form a transition here, and electron transfer happens. And this electron transfer is measured by the transducer, and we can see the signal at the end from the electrode and the signal processor system. So there can be different various ways of electrochemical biosensing. It depends on your design and what your sample type is, what your equipment is, and etc. So you can have a charge displacement, or a charge accumulation, or charge transfer. So there can be various ways to use electrochemical biosensors. So basically, electrochemical biosensors have two separate parts. One is the bioreceptor, and the second one is the biorecognition. As you can see here on this electrode, the little red spot over here is actually the bioreceptor part. This bioreceptor part's material might be composed of different materials. It can be like carbon nanotubes, graphene, or some metallic nanoparticles such as gold, silver, platinum. It can be silica, organic polymers, nanowires, and etc. On this material, we immobilize a biorecognition molecule. It can be an antibody, an enzyme, it can be microorganism, DNA, it can be a whole cell. And I can say, I will talk about them later on, like aptamers, molecularly imprinted polymers, and so on. So whenever the analyte comes and binds to its target here, there has to be an electron transfer, either from the molecular interaction between the analyte and the target, or you have to design either a fluorescent dyes presence, or an enzyme presence, or something else that will start the electron transfer. And this electron transfer signal is processed by the biorecognition part. So it processes the signal and brings out a readable output. So as I said, I will try to give you some information about what types of biorecognition molecules can be used in biosensors, and not only in electrochemical biosensors, but also in most of the biosensors too. So let's start with antibodies. Antibodies are protein components of the immune system that circulates in the blood and recognizes foreign substances. As you can see here, antibodies are composed of two light chains, two heavy chains, which are connected by disulfide bonds, and it forms a V shape here. And the bottom part is generally majorly the same between the same species, and the top parts are called as the antigen binding sites. And it is the variable region. It changes from antigen to antigen, and each antibody have a different antigen binding site. So how antibodies are produced? So basically, it starts with the immunization of of an animal. So with the antigen, so we immunize the animal with the antigen and wait for the immunization response. When we have the immunization response enough, we take out the spleen cells and we fuse them with the tumor cells. The tumor cells are immortal, so when we fuse them together, they become hybridomas right now, and they can produce the antibodies, and they are immortal right now, and we can grow them in cell culture for theoretically forever. And these cells are producing the antibodies that we selected against the antigen, and we can continue here by producing the antibodies and so on. We can immobilize those antibodies on the top of the electrodes of electrochemical biosensors. So the design on the top is depending on what you want to measure. It can be a sandwich model. It can be some other types of designs. You can add a fluorescent dye, you can add an enzyme, and like as in the one here, and put the substrate and start the electron transfer and so on. And we know that antibodies are mainly used in ELISAs, but they're not very like on-site or rapid methods. And as I mentioned before, antibodies are mainly used in lateral flow test strips. And this is a very neat example. This study, they developed an antibody against porcine protein, and it very nicely worked only in the pork extracts, but on the other animals extracts, they don't have any binding. And this is the one that I showed you before from our study. So let's move to the another type of biorecognition molecule. They are called aptamers. Aptamers are short single-stranded DNA or RNA molecules. They can selectively bind to specific targets, including proteins, peptides, carbohydrates, small molecules, toxins, and even live cells. They're actually very similar to antibodies, but antibodies are in the form of proteins, but aptamers are in the form of DNA or RNA. And actually their selectivity and specificities are very similar to antibodies, and sometimes aptamers are having better specificities too. They have the single-stranded DNA or RNA version, but they have the tendency to form helices or single-stranded loops. So rather than the primary sequence, aptamer binding is determined by its tertiary structure. So the target recognition and the binding involves the three-dimensional shape-dependent interactions, as well as hydrophobic interactions, base stacking, and intercalation. So how does aptamers are developed? We start with a single-stranded DNA or RNA library. So these libraries contains various sequences of DNAs or RNAs. For example, if you want to have a 40 base pair DNA library, so for each base, you can have one of the 4 options of adenine, guanine, cytosine, or thymine. So it will be like 4 over 40 times different sequences will be in this library. If you immobilize your target on a surface and add the library in and make an incubation, and then you have to discard the non-binders, and then you have to elute the binders, then we should amplify the binders by PCR. We're going to have an enriched oligonucleotide pool at the end. So this pool we're not going to start over because this is the library, but we will repeat almost the same procedure from this enriched pool of candidate aptamers. So these aptamers are again bound to the incubated with the targeted molecule, and the non-binders are discarded, and the binders were amplified by PCR, and these steps are repeated again and again. And at the end, we have a candidate aptamer, and we sequence it since it's a DNA or RNA, and find the best aptamer for the targeted molecule. So actually, we had an aptamer study too, and we used it as an electrochemical biosensor design. I developed an aptamer against porcine fatty acid binding protein 5, and the sensitivity of this system was between femtogram to picogram per milliliter levels. So that means it was actually, the sensitivity was less than ppb levels. This is the equipment that we used for this electrochemical biosensing. So this is the electrode, and we immobilize the aptamer on it. And this is the signal that we were getting. So if you zoom here, you can see that the yellow graph belongs to before the sample addition. And then with the increasing concentration of porcine protein, the yellow graph moves, the yellow peak moves from pink to purple to red to blue peak. And you can see the difference between yellow and blue, and this is a very nice signal that we can obtain. And it is a biosensor that can make a detection at the contamination levels. So let's move to the last actually biorecognition molecule, which is called as molecularly imprinted polymers. We call them as MIPS. They're plastic antibodies, or they're called as synthetic receptors. So they are actually very similar to antibodies again, but they are formed by chemical reactions. That's why they are called plastic. They start with a template molecule. They add functional monomers and let them to assemble on the template molecule. So each monomer find a place, a surface to bind, and then they add the cross-linker for the polymerization, and they remove the template, and this is the MIP that they form at the end. And here is a study that they developed MIPS against porcine serum albumin. And they did the same procedure, and after the template removal, they had a post-imprinting modification by the addition of the fluorescent molecule, and they immobilized the MIP on gold nanoparticle, and at the end, they put the MIP with the gold nanoparticle on the surface of the electrochemical biosensing equipment electrode. And they see that when the porcine serum albumin is bound to the MIP, it gives the fluorescent signal, but bovine serum albumin cannot be bound to this MIP and doesn't give any signal. So it's a very nice study. So finally, I would like to talk about some optical biosensors. There are various types of optical biosensors, but I would like to focus on vibrational spectroscopy. They're also very rapid, sensitive, and simple, but most importantly, they are non-destructive and non-invasive. They're actually available portable vibrational spectroscopy devices around in food control analysis, and I believe they will be maybe like we will all have them in our hands in the recent future. So vibrational spectroscopy deals with the interaction between electromagnetic radiation in infrared region with molecules. It is the analysis of molecular properties based on vibrations at molecular level, and it is associated with the chemical bonds in the sample. There are different vibrational spectroscopy can have reads at different ranges in the light spectrum, such as infrared, mid-infrared, far infrared, and Raman spectroscopy. So let's start with Ftir spectroscopy. So in a study, they had the Ftir spectra of lard, and they have this nice spectrum. And in another study, they compared it with the beef fat and find out the differences between the beef and the pork. And actually, like each wave number is specifically indicating the functional groups responsible for the absorption. So it is evaluated this way. And at the end, with the help of the statistical methods, all these spectrums are becoming meaningful. So actually, FTIR equipment is not a very small equipment. It is a laboratory method. But after developing a laboratory method and finding out the evaluation procedure, the statistical method, and what wave numbers to focus on, what are the wave numbers that differs, for example, pork from bovine, we can move to the portable devices. You can see portable FTIR devices here. So these are a little bit bigger than some other available biosensors, spectroscopic biosensors, but they are also highly sensitive. So let's move to another optical biosensor, which is Raman Spectroscopy. So you can see that in this study, they differentiated pork lard from other animals' fats, and the difference between Raman and FTIR is the spectrum, the light spectrum that they use. So that means the molecular interactions at those wave numbers will be different between Raman and FTIR or some other spectroscopic methods. So their specificity might differ. So you can see like they look visually kind of similar, but there are actually like some differences between all these graphs. So they have to be statistically evaluated. So they have the peak areas and find out the differences and statistically evaluate the results. And also they studied the percentage of lard in other meat mixtures, and at the end they have a very nice model. So I should say that Raman spectroscopy is also has a huge instrumentation. This one also has a microscope on it, but as I said before, like after the laboratory work, when we know that what wave number is or what type of statistical information we need to have, we can move on to the portable devices. And as you can see, this type of optical biosensors are can measure outside the bottles, outside the packages, and this is a very nice property of optical biosensors. So as the future perspectives, we should focus on from the consumer's perspective and the production factors production perspective. So I should tell you that the types of biosensors are really diversified, and there are many more biosensors that I wasn't able to sequence in this talk, like such as electronic nose, microfluidics, and so on. But I believe this talk can give you some idea about what can be done outside the laboratories and rapidly. And this will help consumers to be their own auditors all around the world, not only in Islamic countries. And also, like, I believe that factories are required to have biosensors placed in the production facilities for the control of the process. So in this way, the consumers will also be able to check from their phones or like some applications what is going into the factories and what is coming out and the whole process. So it will be much more reliable. And I believe this is the future though. Finally, I would like to tell you that what we want is taking the analysis outside the laboratories. And this is not actually what we want. This is what the American Army did like 30 years ago, they were carrying their own laboratories inside a huge bag outside. They do the experiments outside the laboratory. What we want is to have lab-on-a-chip systems. We anticipate to have simple and portable, low-weight lab-on-a-chip equipments, both for the Muslim consumers and the producers or auditors for to give the halal certification. So thank you for listening.
Thank you very much, Dr. Isra. You presented very comprehensive and informative lecture on the topic. Now the session is open for questioning. If you have any question, please ask.
Yes, Assalamu alaikum, Dr. Isra. This is Musharraf. Actually, we both just submitted few months before a project under Pakistan Science Foundation and 2B Tech call, and the project is still under evaluation. So, for the So I would definitely congratulate you to highlight such an important issue, and I totally agree with you that that would be the future of halal testing. It must be a kind of on-site testing devices available. My question is that, I mean, do you see, because fairly speaking, so far I have never been exposed such type of halal on testing strips on devices so far. So, I mean, do you see, or there is a commercially available strips for testing of, the non-halal ingredients? If yes, then what about their cost? Thank you.
Thank you, Dr. Musharraf, and thank you for all those nice words. You're a great doctor too. Thank you. Actually, those kind of biosensors are not very available right now. They're, as you have seen from the presentation, most of the results were like scientific literature, but they haven't been outside. But, like lateral flow test strips, like the ones that I showed, similar to pregnancy tests. So they're actually available like for the last maybe 10, 15 years. There are a few companies, not in Turkey though, it's generally like Russian, France, and United States, and so on. Those companies are having this pork specific and sometimes like a large specific lateral flow test strip kits. And one kit is around like $5 to $10 in cost. But like if you want, as I said, like if you want to test the meat, they are good enough. Like you don't need the PCR or something. But if you want to make sure that like whether there is gelatin or some other animal byproducts, which is really hard to get the DNA or proteins out, like I mean like the contamination level, those types of biosensors are not that sensitive. So the sensitive ones are not outside the laboratories yet.
Thank you. Thank you, Dr. Isra. It was a very nice presentation. I'm Hamid from counterpart of 2B Tech, which is in Pakistan, PCSIR. And I am a meat biochemist. And I have been keeping eye on this research. Your field is very, very important from halal point of view. I think this is something, a main point or main issue in this whole halal food business, which needs to be addressed appropriately at a reasonable cost and with reasonable accuracy, like ppm or ppb, whatever it may be. As I understand, part of my question was already made by Professor Musharraf, but anyway, as I understand, these kits which you mentioned are being used for the last 2 decades almost. And mostly these Arab countries, municipalities in UAE and Saudi Arabia, and maybe some other rich Muslim countries were using. But as I understand, their accuracy is about 1% mixture can be detected. I'm not sure, but this is what I heard. But other part of the thing is that work that you are doing, do you see the result of your work or biosensors coming into action at a reasonable cost in about 5 to 6 years' time or so, with reasonable accuracy, which is acceptable to halal industry and halal consumers in the Muslim countries? Thank you.
Thank you, Dr. Mohammed. Nice to meet you. Actually, like this is what we want. Like food generally comes after health. So generally they're for health concerns, those types of methodologies are developed, and they spend all these monies, they can spend all these monies for health reasons. But when it comes to the food, we cannot find that high budgets, So that's why actually the food control systems or food analysis methods, it's hard to find money, So it depends on the money you have, the budgets, the projects you have. But I believe, like as I told you, the glucose biosensors, blood glucose biosensors, have been out like maybe more than 50 years, but we know them maybe like for the last 20 years. They have been common for the last 20 years. So it requires some time, like the consumers have to be adapted. We can develop meantime biosensors, which are very sensitive. And actually, like after you have the research, the production and the cost for one analysis is not that high. Like, and if you compare the analysis budgets, it's like in laboratories, they're really costly because they have all these huge equipments and the man is working and, like all these buildings and everything. So the costs are higher. But when you have those types of biosensors, it's just a little like the science, the research is done. After it's done, the production is not that high cost. It's not that costly. So that means like if we have the research enough, we can actually have them in like 10 years all around the world. Like sometimes they have to these scientific researchers, like they have to reach a peak and then like they will be all around. So I believe like this will be the case in 10 years. Because everyone is having their phones and like want to like put some biosensors or some other like cameras and something on it and want to see the results from the application. So it become very common in the last 5-10 years. So I believe it will move to the food research too. So I am having the same feelings with you. So I believe in 10 years, we're going to have those types of biosensors all around the food control facilities. But what I want from my heart is why shouldn't we be the ones who do it in the halal food control? So we can be the representative for the world food control. So.
Thank you. Again, thank you, Dr. Isra, for presenting a very nice and informative lecture. Goodbye, and thank you to all the Turkish people. Thank you. The next speaker is Dr. Hamida Muhammad. Her topic is current porcine DNA detection method in food and gelatin capsules. Dr. Hamida Muhammad graduated with PhD in biotechnology from Faculty of Science at Mahidol University, Thailand. As a co-researcher, she received Mahidol Award in the research project of recombinant protein production in Escherichia coli for vaccine production. She graduated with Master of Science degree in the clinical biochemistry, molecular medicine from the Faculty of Allied Health Sciences, Chokolam University. Dr. Hamiza has experience in halal science work as a researcher, including halal forensic science laboratory service, halal food verification research, and halal quality system implementation. Her research interests focus on molecular biotechnology and halal science. Her publication titled High Level Accumulation of Soluble Diphtheria Toxin Mutant CRM197 with co-expression of chaperone in recombinant Escherichia coli 2015 reflect her interest in combination protein production in bacterial cell. Please, Dr. Hamida.
Thank you for your kind introduction. Assalamu alaikum wa rahmatullahi wa barakatuh. Today, I would like to present and share our experience in our laboratory service in the topic of current pulsar DNA detection methods in food and gelatin capsule. First of all, this slide show the outline of my presentation today. First, I'm going to present the basic knowledge of the genetics and polymerase chain reaction, including primary size, genomic DNA extraction and purification, and real-time PCR for food and gelatin samples that suitable for routine laboratory service. And finally, I gonna present of our laboratory's experience in recent situation. Living organisms are divided into six kingdoms: animal, plant, fungi, protists, eubacteria, and archaeobacteria. Every organism is different genetic substance called DNA. The different DNAs mean the type and sequence of the DNA base, which are responsible for the difference and diversity of living organisms. These difference they enable to identification of each species in living organisms. And genes are the fundamental units of heredity, and they determine specific characteristics of an organism. Gene made of nucleic acid and molecular and biological molecules that are found within the structure called chromosome. In many organisms, genes are located on chromosome in the nucleus of the cell, in every cell of individual's body. In 1953, James Watson and Francis Crick announced the discovery of DNA structure known as double helix. It consists of backbone of the double helix. And DNA consists of long chains of alternate units of phosphate and deoxyribose sugar. The DNA double helix is stable by hydrogen bond between the base and attached to the two strands. The four bases found in DNA are adenine, glycine, cytosine, guanine, and thymine. The Human Genome Project started in 1990. The international scientific research project with goal of determining the base pair that make up human DNA and identify and mapping origin of the human genome from both physical and functional standpoint. And the Human Genome Project complete into 2003, and then the swine genome sequence we will obtain, and then Pig Genome Project was performed in the swine genome sequencing consortiums. This slide shows that all species in this family originate in over Europe, Africa, and Asia. This slide shows the pigs in 19 species in the family of in the family of Sudae. The pigs that most people familiar with domestic pigs. Almost of these pigs were domestic from Saskova or Weiber. Everyone can know the polymerase chain reaction is the gold standard in the diagnostic testing. The polymerase chain reaction is one of the most powerful technology in molecular biology. And using PCR specific sequence within a DNA and cDNA template can be copied or amplified many thousands to million-fold using sequence specific nucleotide. PCR is the greatest important for the purpose of species identification. Several methods based on PCR have been proposed as useful for identifying species of origin in foods because of its high specificity, high sensitivity, rapid processing time, and low cost. This technique invented by Kelly Morris in 1944. In 1993, his method became a central technique in biochemistry and molecular medicine. PCR is the in vitro technique for generate large quantity of specific DNA sequence. In a simple word, ultimate version of DNA replication is the consist of 3 steps of the PCR cycle. And double strand DNA were heat at the 94 to 98 degrees Celsius. The double strand were separate to the single strand DNA. And annealing the primer bind to the primer of specific primer and bind to the target of DNA. And the lock in and then elongation, the extension of the primer sequence over the target segment with the DNA polymerase. These steps are repeat 25 to 35 cycles to exponential produce exact copy of the target DNA. What is the primers? Primers is a short synthetic oligonucleotide which is designed to complementary of the region to the target DNA. In this case, specific to the porcine DNA, it consists of forward and reverse primer. Primers, the good of primers should generally have the following properties. Length DNA base about 18 to 24 base and contain 40 to 60% GC content and start and end with 1 to 2 GC pair. Melting temperature of 50 to 60 degrees Celsius. Primer pair should have a TM within 5 degrees Celsius of each other. And primer pairs should not have complementary regions. When we can retrieve the Saskova nucleotide sequence from NCBI or GenBank, and we choose the regions of mitochondrial DNA that serve a model of the genome evolution. This slide shows the primer design program are available online platform. In case of genomic DNA extraction and purification, consists of 6 standard methods such as boiling, centrifugation, phenochloroform, lysis reagent, spin filter columns, magnetic bead technique, and automatic extraction. And genome DNA extraction method isolate genomic DNA away from protein, DNA, and other cellular materials. And method to isolate DNA from the food can be complicated because of carryover of food component that may inhibit PCR reaction. This slide shows the commercial DNA extraction kit for food example are available on the market. And after DNA extraction, we will perform PCR, and PCR component consists of tag DNA polymerase, a primer, DNTP buffer, and DNA templates as obtained from extraction. And conventional PCR has detection at the endpoint of the reaction using gel electrophoresis. And gel electrophoresis is the technique used to separate DNA fragment according to their size. A real-time PCR instrument platform consists of the thermocycler optic for both fluorescence excitation and emission collection, together with the computer and software analysis. In the reaction, we must add the fluorescent dye or fluorescent probe to monitor the progress of amplification reaction. The most sensitivity and responsible method of real-time PCR measure the fluorescence at each cycle at the amplification progress. This allows quantification of template to be based on the fluorescence signal during the exponential phase uh, of the amplifications. This slide shows the uh, two methods of the fluorescent dye and fluorescent probe in the real-time PCR. We focus on the cybergreen and hydrolysis or Tagman probes. For the cybergreen, DNA bind to the double strand. When the DNA denature the cybergreen dye flow free, as shown in this figure B. And when cybergreen dye bind to the double strand DNA, as shown in figure C, the intensity of fluorescent emission increase. As the more double strand amplicon are produced, cybergreen dye signal will increase, as shown in the figure D. For the hydrolysis probe or the Tagman probe, with fluorescent reporter at the 5 palm end and the quencher at the 3 palm end. Commonly used fluorescent reporter quencher pair as a fluorescent FAM and emit green fluorescent and black hole quencher dye. In step 1, during primer annealing, hydrolysis probe bind to the target sequence. The fluorescence of the reporter is quenched due to its proximity to the quencher. In the stage B, during extension, the probe is partially displaced and the reporter is cleaved. As a result, the reporter is separate from the quencher, result in the fluorescence signal that is proportional the amount of amplified products in the sample as shown in stage B. The next slide, we show the equipment and reagent for the genomic DNA extraction and real-time PCR in our laboratory. Detection of pulsar DNA from food products by real-time PCR consists of 2 steps, and DNA extraction of in food products and amplified DNA target with real-time PCR. This is the lab where equipment and the extraction kit from Promega and check-in incubator, microcentrifuge, vertex mixer, and spin out. And pulsar DNA of the sample were amplified by using real-time PCR as similar to positive. Pulsar DNA of the sample were amplified as shown in the, okay, sorry. The amplification pulsar DNA of the sample were amplified as shown in the S-curve, similar to the positive controls as pulsar DNA. Okay. This video is the demonstrate about the DNA-based lysis solution and the real-time PCR-based cybergreen fluorescent dye. โอเค it's not move sorry for เดี๋ยวนะคะนี่ค่ะ
sorry แป๊บนึงค่ะ
stop share ครับเดี๋ยวนะออกไปที่สกรีน
การตรวจ DNA คือตอนหนึ่งสารพันธุกรรมคือตอนในผลิตภัณฑ์อาหารนะคะโดยใช้เครื่องจักรทำ PCR ในการวิเคราะห์ตรวจหาปริมาณการปนเปื้อน DNA ในผลิตภัณฑ์อาหารผลิตภัณฑ์ส่วนใหญ่ที่นำมาตรวจวิเคราะห์ในทางด้านละเอียด DNA นะคะจะเป็นผลิตภัณฑ์
ค
Dr. Hamida, can you listen me? Hello,
Hello.
can you
Wait a
listen me?
minute, please.
Did you finish? Actually, we were not listening to you. The voice was not coming.
ยังค่ะไม่ใช่
Did you finish? Hello,
Okay. Sorry.
you finish your
I
lecture?
will continue my slides. Okay.
Okay.
Okay. Okay, in case of gelatin capsule and gummy candy, we will perform the method similar to the detect in food sample, but okay, in case of gelatin capsule and jelly, we will perform the method similar to the detection in food sample, but the DNA extraction from gelatin containing food is more difficult. This slide shows the method gelatin. Okay. This slide shows the method gelatin extraction from tissue containing collagen, such as alkali process and acidic process. Gelatin and gelatin-containing food generally contain very low amount of highly degraded extracellular DNA. This fact make the isolation of DNA more difficult. And gelatin-containing food generally contain very low amount of highly degraded extracellular DNA. So the extraction DNA is insufficient quantity for real-time PCR by increasing sample amount to 2 milligram or 1 gram sample amount. And these two methods were used to extract gelatin sample both of capsule and gummy. I used to extract gelatin containing food from both commercial kit. And then this slide shows current our research project, multiplex HRMA based on real-time PCR technique to detect forbidden animal contamination in halal food products for laboratory service. And development of a strip test based on nucleic acid lateral fold to detect forbidden animal contamination in halal food products for on-site consumer service. In the next slide, I would like to share our experience, our laboratory service that I never thought would happen during COVID-19 epidemic. This figure shows Dr. Vinay warned people about pig fake as a cow, as shown in the poster behind him. Under situation COVID-19 epidemic, food delivery service is booming. Our laboratory service received boiling meat for detection of the porcine DNA. Sender said that he bought meat from seller as a Muslim and delivery meat to his home. When he boiling meat, the color of meat are different from previous cooking. After that, the scientists analyzed meat sample with the real-time PCR. The results show the porcine DNA when using porcine primer and present positive of bovine DNA when using bovine primer. But the positive of bovine show lower amount of DNA than positive of porcine. So we demonstrate pork dipped with cow blood and detection with real-time PCR. The results show the same as previous suspected meat. It concludes that suspected meat is pork contaminated with cow blood. After that, more than 40 meat samples sent to the HSC laboratory service for testing porcine DNA. All of meat that you see, the results are pork contaminated with cow blood and beef contaminated with pig blood. When we trace back to the meat sample, all of meat sample without halal certified, but seller as a Muslim. The main Muslim consumer has trust seller as Muslim, regarded of Halal certified. And the other reasons of COVID-19 epidemic, people layoff have become so widespread. Food delivery as an option job for Muslim. So meat sample pass is on each seller, which they don't know the source. And finally reason, price of beef is more expensive than pork. As a result, adulteration pork into beef more profit. We conclude that Halal forensic science is the important role to support the scientific information for solve the problem. This slide shows the overall sample of 4 analysis in 2004 to 2019, including DNA, gelatin, alcohol, and fatty acids that Dr. Natchawan mentioned previous. And analytical technique of HSC for non-halal material were published on the Halal Food Production books and also published on Journal of Food Protection. And I would like to invite our audience for participating Thailand Halal Assembly 2021 in the concept of a virtual way for actual halal world. It will be here on 14 to 15, 2021. This is all of my presentation. Thank you.
Thank you, Dr. Hamida. A nice presentation. Do we have any question to ask from Dr. Hamida? Thank you, Dr. Hamida. Due to shortage of time, we will skip the question and answer session. Thank you so much.
Thank you. Thank you very much.
Thank you very much. The next speaker is Dr. Shakil. He is from the Panjwani Center for Molecular Biology. Dr. Shakil, his topic is a simple and sensitive NGS-based method for pore detection in complex food samples. A little bit introduction about Dr. Shakil. Dr. Shakil obtained his PhD degree in molecular medicine from University of Karachi. He's working on next generation DNA sequencing for the last 7 years. His work involved molecular basis of diseases through genomics. He is also doing molecular biological aspect of pore detection in food sample. Dr. Shakil.
Bismillahirrahmanirrahim. Assalamu alaikum to everybody. Thank you very much for inviting me for this lecture. The lecture series are involving a lot of latest technologies. The emergence of these technologies have enabled scientists to analyze the data with more confidence to find the desired results. In my talk, I will cover briefly the next generation DNA sequencing and how it can be applied in halal food testing. So basically the objectives for this lecture would be what is DNA sequencing and what is next generation DNA sequencing, and how these techniques can be utilized to generate data and analyze, and finally to find the species which may be present in the food sample. So I'm not going into the details what is the DNA, because a lot of speakers have told already so much. So briefly, DNA is a polymer of 4 types of nucleotides which exist as a double-stranded form in living organisms. So they exist in a way that one compound, which is adenine, is always complement with another compound, that is thymine, and vice versa. And cytosine always complements with another base, that is guanine, and vice versa. And this one pair is termed as base pair. So this terminology, that base pair, is frequently used in the genomics. Now, the overall content of DNA in one cell of an organism is termed as genome. The good thing about genome is that every species has specific number of nucleotide bases in its genome. For example, in human, there are approximately 6.4 billion base pairs which are arranged in 46 chromosomes. These 46 chromosomes are basically large DNA molecules. Likewise, in domestic cow, there are 2.86 billion base pairs of DNA which are arranged in 60 chromosomes. In a pig, there are 2.58 billions of base pairs of DNA which are arranged in 38 large molecules of DNA. So what is DNA sequencing? Very briefly, it is basically the reading of the sequence of nucleotide bases in a DNA molecule. So there have been different techniques to determine the sequence of nucleotide bases in a DNA. Initially, a Sanger sequencing was very long ago in 1980s, and then recently the technology has advanced and second generation and third generation sequencing techniques have been developed. These second generation and third generation techniques are termed as next generation DNA sequencing technologies. The difference between first generation and next generation techniques is that in first generation sequencing technique, we can only sequence a small piece of DNA in one direction, while in next generation sequencing, we can determine the sequencing of almost whole genome of an organism. So in Sanger sequencing method, what occurs? We start with one type of DNA molecules or one type of DNA strands, which is approximately 100 base pair long. So we produce many copies of this DNA strand which are exactly differ in one base pair, right? Then we pass these newly generated DNA copies through a capillary which separates based on this size. During this separation, each base is detected, and in this way, the sequence of a short fragment of DNA is determined. But in next generation DNA sequencing, as I have told, the complete genome of an organism can be sequenced in one reaction. So, how this is accomplished? This technique uses flow cells. These are basically small chips. This chip is approximately 2 to 3 centimeter wide and 5 centimeter to 6 centimeter long. So this contains a lot of nanoreactors where the DNA fragments of approximately 300 to 500 base pairs are loaded, and the sequencing reactions are performed. Basically, millions of sequencing reactions are performed, and in a way, we can determine the sequence of nucleotides of the whole genome. So what kind of data is generated in next generation DNA sequencing. So unlike the other techniques where different types of chromatograms are produced, here a text format data is generated. So one read is represented by 4 lines. So first line is a unique identifier for one DNA read. The second line is the DNA sequence. The third line is a plus sign, and the fourth line is the quality score of each sequenced base. So these 4 lines are repeated again and again for millions of times, which represent the DNA sequences of millions of short DNA reads. So now I should explain briefly how NGS can be applied in halal food testing. So the basic principle is very simple. Genomic DNA is isolated from the food sample. The DNA is sequenced, and then the generated data is matched with the available data which is available in online resources. So the whole process can be divided into 4 steps: DNA isolation, and second is sample preparation, which is also called as library preparation. The third is DNA sequencing using NGS platform, and the fourth is data analysis. I quickly and briefly go through these 4 steps. In the first step, a specific quantity of food sample is crushed in a liquid nitrogen, and then using a commercial kit, the DNA is isolated. As we have developed a program, so we use this commercial kit which is produced by QIAGEN. So, but literally any kit can be used which can provide good quality of DNA. For DNA quantification, a fluorescence-based method is used, which only determines the quantity of double-stranded DNA. For performing sequencing on next-generation sequencing platform, the DNA should be of high purity. The absorbance at 260 to 280 ratio should be in the range of 1.7 to 1.9. Then for the library preparation, there are a number of kits available, but I have quoted here the most widely used kit, which is Illumina DNA Prep Augmentation Kit. What happens? Basically, the genomic DNA, which may be 200 million base pair long or even more, is reacted with transposomes. These transposomes cut these DNA into approximately 300 base pair molecules, and also add some adapters which have known sequences. So this process is termed as tagmentation, because the fragmentation of DNA and addition of tags takes place in a single step. So as a result, this genomic DNA has been transformed into millions of short DNA molecules which have short oligos attached to the both ends. Then in next step, we add another adapters which term index 1 and index 2 or P5 or P7 adapters. These are unique identity. This is used when we have to multiplex more than one samples in one reaction. So after adding these indexes, the sequencing ready DNA fragments, which we term the library, is ready for sequencing. So before sequencing, we have to determine the accurate quantity of the DNA molecules. For this, we use this formula, and we determine the nanomoles of the library. For performing sequencing, it is very necessary that we use optimum amount of libraries so that a good quality of data is generated. Normally for sequencing on the Mysq platform, we use 10 to 12 picomoles of library, which is added to the Reagent cartridge, which is provided by the manufacturer. So this is basically the Mysq platform. This has 3 components which we interfere. The first part is where we load the flow cell where sequencing is performed. There is a second part. Here we load the sequencing cartridge, which contains all the chemicals and reagents for performing the sequencing reactions. And this is a C chamber where we load the wash buffer, which is also consumed during the data generation. So this is an screenshot of a successful Mysq sequencing run. So this graph shows the quality of data. So here, this represents millions of DNA reads which have been sequenced, and this shows the quality score of the data. So for good quality score, as I have told, the quality should be more than 30. Normally the quality score ranges from 0 to 40, but greater than 30 is the acceptable range of the data. So next, once the NGS data has been generated, we have to match this data with the online resources to determine the species which might whose meat may be present in the food sample. For this purpose, we use the vastly used BLAST program. So I have also given the basic commands which one can use on a Linux platform. So this is a few lines which we get after BLAST analysis. So this is the identity of a DNA molecule which was sequenced, and this is the ID of the subject for which this sequence matches, and this gives the general name of the title for which this DNA molecule matches. And there are also certain quality scores like percent identity, number of identical bases, mismatches, E-value, and the Q quality per HSP. So for good analysis, the percent identity must be 100%. There shouldn't be any mismatch. So mismatch must be 0. And this value, Q coverage per HSP must be 100. E-value should be very low. We have optimized with 10 to the power minus 10 E-value. Here you can see the E-value is approximately 10 to the power minus 29, minus 29. These values are extremely low, which means that the identity between the sequence DNA molecules and the sequence which is present in the database is very, very high. So these parameters must be considered for performing these analyses. So we have published this protocol recently in the Reagent Journal of Chemistry. So briefly, what we did indeed, we prepared 5 samples. 4 samples were port positive and 1 sample was port negative. We prepared actually complex food samples in which the cooked food and uncooked food also present. You can see the finch feather, fish gills and mussels and many things were added to the samples to make it more and more complex. Then after the sequencing, we obtained these number of DNA reads, and after the BLAST analysis, for sample 1, the spike proportion was 0.15%, and the proportion which was detected through the analysis was 0.14% approximately. Sample 2 was negative sample, which didn't contain port spiking, so it didn't detected any DNA reads. For other samples, the spike proportion and the observed proportions are nearly close. So we also compared our method with the previously published methods. So basically this method is applicable to those samples where PCR cannot be performed due to because the sample is highly processed or degraded and PCR gives negative results. So by performing NCS, we can detect the source species whose meat is present in the sample. So to conclude, DNA, the next generation sequencing can be applied to testing the adulterant non-halal meat in the complex food samples. So this method have been developed to determine the contaminants qualitatively. So we are further working on this project and will optimize to determine the adulterant quantitatively and also for accreditation. So this is our Jamilur Rahman Center for Genome Laboratory at the ICCBS University of Karachi, where this work was performed. And thank you so much. And you are welcome to ask questions. We actually, for this procedure development, we performed 5 samples. Yes. Yeah, this is first report of this method. So,
Any other questions?
Assalamu alaikum.
Walaikum assalam.
Dr. Zishan, as we know that we cook food at a very high temperature, that could degrade the DNA. So what do you think that how might this method could be accurate for cooked food?
So in our method, we also used cooked food in the sample. So I think also DNA is degraded, but there are fragments of DNA which may survive. So if minute quantity of DNA or short nucleotides is available in the sample, this could be detected through this method.
Thank you.
Yes. Yeah. At this, thank you for this question. This is very important question. This process takes, if the protocol is optimized and things are working perfectly, so the work which I have presented, this will take 2 days, 2 full days, right? And for the cost, yet NGS is costly at this time, and we can expect that with the passage of time, the cost will be reduced and is applicable. Basically, this method is applied only for complex foods where the cheap process of a PCR cannot be applied. So this is the price of this test, that you can detect the sample which cannot be detected using routine methods. So that's the advantage.
Assalamu alaikum, sir.
Walaikum salam.
Very nice talk, Dr. Shakil Ahmed, sir. My question is, when you blast the data library and data in the NCBI, you have mentioned E-value. What kind of information you extracted from E-value?
So E-value basically is the probability that our sequence, what is the confidence that our query sequence matches with the subject, right? If the value is quite low, then the probability is that this matching is not by chance or due to any error. This is genuine matching, right? So briefly, this is what E-value means. The lower the E-value, the higher the confidence of matching our query DNA sequence and the subject DNA sequence.
Dr. Shakil, thank you for your precise talk.
Thank you.
Please, may I know how do you get the primers for DNA sequencing?
Actually, we do not need the primers. Actually, I have quoted this kit, Illumina DNA Prep Segmentation Kit. So this kit contains all the consumables for library preparation, right? And for also sequencing, we also use the predefined kits. We do not need any primers or things like that.
Thank you.
Yes.
The cost of this.
At present, the cost for one sample is approximately $150 or $200. So again, cost is dependent on the number of samples. If we are doing less number of samples, in this case we have performed 5 samples. So the cost per sample was approximately $200. But if we are performing large number of samples, like 100 samples or 50 samples, then the cost will be reduced to approximately $50. So this is the typical aspect of NGS. The larger the number of samples, the lower is the cost. Thank you.
Assalamu alaikum, sir.
Any question?
Yes, please.
Yes.
First of all, very nice talk, sir. I want to know that NGS, you are going to perform whole exome sequencing or whole genome sequencing?
So this is whole genome sequencing basically, because for performing whole exome, we need certain probes which are species specific. So also for performing exome, if the food sample is highly processed, there are highly chances that we might not be able to enrich the exomic part of the genome, right? So that's why it is recommended that we go for the whole genome. So it is not the intention to cover the whole genome. If we only recover 200, 300, or 500, or even 100, 1000 regions of the genome, so that would be enough to identify the species which we target.
Thank you so much, sir.
No question. Thank you, Dr. Shakil, for nice elaboration. Our last speaker is Dr. Nuratni Binti Ishaq. She's from Malaysia. A little bit introduction about Dr. Nuratni. She received PhD from Sain University, Malaysia. She possesses 13-year working experience, including molecular biology, R&D, work in government organization, and asset procurement on ISO documentation. She has been appointed a technical trainee editor by Department of Standard Malaysia in conducting molecular biology testing to meet ISO 17025 and ISO 9001 requirements. She has very long list of experience. So due to shortage of time, we will request Dr. Nuratni to proceed, please.
Okay, Assalamualaikum. Did you hear me? I'm the last presenter for today. Are you hear me? Yes. Okay. Okay, I'm the last presenter. So in Malaysia right now, we are 8 p.m. It's nearly to our bedtime. I'm sorry. Maybe because of our time have differences of the time. Okay, so my I just want to share with all of you my research. It's currently hit up in Malaysia. It's about the frog meat in Malaysia. Even though it's not involved porcine or pork, but then in terms of slaughtering and everything. So my presentation very short and simple. So now I will sharing my presentation with you all. Mana? Apa? Apa? Okay. Macam mana nak tengok? Okay, did everybody can see my presentation, my slide? Hello, anybody can see my slide or cannot see my slide?
Yes, yes.
Okay. So I continue. Actually, my research is about identification of deer's DNA, which is in Malaysia, they are selling actually the cow meat but they claim as deer meat. Actually, deer meat very expensive. It's cost 100 over per kilo. Compared to the cow meat, is only 30 ringgit per kilo. Okay, so I'm Noratini Isha. I'm from Department of Chemistry Malaysia under Ministry of Science, Technology and Innovation. Okay, so I continue. Okay. So this, I just want to tell everybody what is the Department of Chemistry. Actually, Department of Chemistry is a provider agency, which is we have a non-forensic and forensic division. And then actually we do a lot of testing, which is we do analysis, consultations to the government agency, enforcement agency and also to the private sector in terms of analyzing their samples when they need a consultation. So we will go and consult them. Okay. So okay, Department of Chemistry actually is aged more than 100 years since British time. So each of the state in Malaysia, we have branches. But the HQ was in Petaling Jaya, Selangor. Okay, each state we have, but for the DNA analysis only in Petaling Jaya, Kuching, and also Johor. But for halal testing is only in Petaling Jaya. Johor and Kuching is more to the forensics DNA. Okay, so in Malaysia, you know, there's no problem to send sample either for the private sector, government agency, enforcement or anybody. Just they can find the nearest branches at the state in Malaysia, so they can send sample, follow the procedure as per decided by the department, my departments. Okay, so we goes to my research. Okay, actually the previous speaker had mentioned about DNA sequencing, but my research is very simple. Okay, I just stopped my study in DNA sequencing. Actually, the benefit of DNA sequencing is for gene isolation, for forensic sequence categorization, gene interaction, and also cloning. But what we adopt at Jabatan Department of Chemistry Malaysia most to forensics, more to forensic and non-forensics. Okay. Before this, before we have sequencing, we go to identify the species. We use key binomial, which is you have to see the fur, the blister. You have to see the thorax, and then you have to compare with the key, which is was put in one book. Okay, but after the inventions, everything, the scientists go for the molecular, which is from the specimen. The extraction just only take 1 day. Okay, and then you do the PCR only 1 day. After you extract the DNA, we do a PCR amplification, and then after that, there are a few steps like cycle sequencing, purification, and then you just put in the genetic analyzer. There are a lot of the version, the type of genetic analyzer. So at our department, we use 3500, we have 3130. Most of the genetic analyzer machine is from the ABI. Okay. So before this, we have to use microscope. So when we apply the sequencing approaches, sequencing assay is only take less than 1 week, less than 5 days to give you a result. Compare the conventional method, which is more than 7 days. You have to take times to identify the species. Okay. So the benefit of the sequencing assay is you can determine what is the new breed, the good DNA that you can compile and put in the database so that you can use to breed a new species. Okay, but that is not a lab modification. It means that might be just like you want before this, like I go to training in the India, they teach me from the sequencing you can identify like cow which is have more muscles compared cow have more fat. So of course you as a breeder you want more a cow which is more muscle compared fat because it give you a more profit. Okay, sequencing also help you help the government agency in to aid the fraud cases. Like in my study was a deer meat. Because why? Before you slaughtered the deer, you can see the physical is like the physical was a deer, but after you slaughter, you know, when you packaging the meat, you cannot identify either the meat is a cow meat or deer meat. So in order to identify what kinds of the meat they're selling in the market, so DNA approaches, especially for DNA sequencing, is the best method to identify because from the sequencing you can know the location, you know the breed, you know the primer you use. Okay, that's good. Okay, and then besides that, it helps in zoonotic disease. Okay, because from the sequence of the DNA you get from the sequencing is sequencing technique, you can see where is the mutation, the deletion, insertion. Okay, and that is good also for the animal database. For examples like for the wildlife samples, you know, actually deer or I can say is a service species. There are two types. One is service or deer that you breed in the farm. One is a deer that a wildlife. Okay, in Malaysia, actually the wildlife deer you cannot just go and just go and shoot and then and sell. It's illegal. So if you been caught by the police, okay, when you go to shoot the deer, so you can go to the jail. Okay, compared to the deer that you breed in the farm is different because you are purposely breed the deer for to sell to the public. Okay, so this is important. The sequence DNA that we get from the sequencing analysis, we can build up the database for the deer, for any animal. You can apply that. Okay, because I think all over the world you can find there are a group of person of individual who like to eat the exotic meat. Okay, like deer meat. Okay, sometimes I also receive the pangolins. Okay, so the sequence analysis is good for DNA for the animal database. Okay, so this is the flow. The flow to do the sequencing analysis. First, the extraction. Actually, for the raw meat, it's very simple. You can find any available kit. But just like the previous speaker said, for the difficult sample, you might have to modify a bit your extraction method because when you for like raw sample is very easy. So sometimes when they have a raw meat, they like to make a burger, they like to make a sausage. So when you add additional ingredient like flour, you know, gelatin inside, they're very difficult to get your DNA. So the extraction, you cannot round up one extraction method for all sample. So in our lab, we have for raw extraction method, for gelatin, we have for the chocolate, we have the oil, we have different extraction method based on the food matrices. Okay. And then for the sequencing analysis that I apply in my sample for the deer meat, we go through the simple one. I use epicenters, and then I do PCR polymerase reaction. Actually, I have conducted 2 primer. One is cytochrome B gene, and one is one is cytochrome oxidase subunit 1. Okay, between these 2 primer, it shows that cytochrome oxidase subunit 1 is much more better compared to cytochrome B genes. Okay, why I don't want to use a specific primer for Fdir, for servers group? Because why? Servers have a lot of species. For example, they have a timorense, they have a SC, they have a Dama. You know, there are a lot more than 5 species. So it's not practical to go through one by one because it's waste your time. So I decide to find the universal primer, but specifically for servers group. It's much more better. Okay, so because why? If got adulteration, then more than 1 species, so you will get mix of profile. So from there you can separate, and then you can align, then you can blast, and specifically they will tell you what is the species of the servers meat. Okay, after we do polymerase PCR, we conduct purification. This one for we purify the PCR product. For my lab, we use gene oil. Okay, and then as usual, we have to do the cycle sequencing. So from the cycle sequencing, I use primer for PCR 10 picomole. For cycle sequencing, I use 3.2 picomoles. Okay, so cycle sequencing, we use the DNA that we purify the PCR product. After that, I will put in the thermocycler to run another PCR. That's what we call cycle sequencing. During the cycle sequencing, we will divide between forward and reverse. Okay. So after purify, you clean up the cycle sequencing product, and then you put the high dye and everything. Normally, I put high dye after I dry the cycle sequencing product. I put 15 microlit, but sometimes it depends to the instrument. Like 3500, you don't need to dry up your cycle sequencing product. You just can put 15 microlit, and they just put directly to the machine. Okay, so after I purify, I put the high dye, so I just put in the machine, and then I run sometime 4 array and 5 array. Okay, so this is the process. Okay, the time frames to do the analysis, sometimes it can go maximum is 4 to 5 days. Okay, why in my lab might be most of the lab in your country might be lesser than mine, because why? Besides the sequencing, we also have a real time, so we have to occupy and manage our time. Okay, so okay, from the analysis, the deer meat that I do, extraction, cycle sequencing. So I compare between one species, Servus timorensis, but in the 2 different locations, one in the Biluk Pahang, and then one in the Tanjung Malim, Perak. Okay, from the DNA that I get from this same species, but different to individuals, I find out that the maximum identification was a 94%. Okay, because why? There are a few gap here. If you see here, there are a few gap, which is there are these 2 individuals, even though they are same species, but they carry their own DNA. Okay. So after I investigate why it happened. So the factors of their diets, because in the Biluk Pahang is like at the Cameron Highland, very cool place, So the DNA, might be this is just my assumption, might be the adaptation to the environment, and then I ask the breeder what you give to the deer. Say they use a very organic diet compared to the in Tanjung Malim. So, but then that's just curious me, and then I do investigation. But then it doesn't says that what you eat will change your DNA. It takes a long time. Maybe the adaptation sometimes. Okay. Okay. So from the analysis, this one I use cytochrome subunit 1. So the maximum identification 94. So when you involve in the sequencing, it should be more than 94%. Okay, maximum identification. Okay, so when I blast through the NCBI is a very general for blasting the sequence DNA. So from here, they are to get which is from 100%, it goes to 94% maximum identification. From the analysis of the sequence DNA I conduct, so it found out it's a Rusa timorensis or Cervus timorensis, and then it's originate from the Bali, Indonesia, and then yes, it's a mitochondrial, then it's a partial genome, it's not a complete genome. Okay, then from the analysis, this from the DNA sequence that you get. Okay, this is the result that I get. Okay, sorry, the picture was not very clear. Okay, so from there I do the sequencing analysis. I align with the software. Actually, to align the software, you can easily get from Mega, Pulp, Philip, but then for me, I use the free one and easy to download from the internet. So I use Mega app. Pulp, Philip, Puzzle, you have to pay because it's I think in Ringgit Malaysia is more than 5,000. Okay. And then after I align, okay, first step, I get the result. And then I align using Mega X, the software. Okay. So from here, you can see if you get the similar DNA, I mean, it match each other, okay, it will give you like G, G, G. So they are here A different. Sorry. So here A is a different. Okay. This one for this analysis, I use 3 individu from Biluk Pahang and 3 individu from Tanjung Malim. So after I align the DNA sequence that I get, I to do the populations, I compute the sequence of DNA that I align using I use the P distance. P distance is for genetic distance. Okay. So here there are software here. So you can choose either you want the P distance is for genetic, you want Tamurane, you want for phylogenetic, you can choose. But it's not simply for genetic, you genetic distance, you use P distance. And then for phylogenetic tree, you use Tamurane. No. If you start from P distance, so until the end, the analysis for the statistic is for, you must use P distance. So after I compute the genetic distance, you can see here, the Excel was not very good. Okay, you can see each of the species give a value of your genetic distance. Genetic distance means that if you have a very small amount of the differences, means that you are very close to your species. Okay. After that, after I get the genetic distance between 6 individu, I want to construct the phylogenetic tree. I want to see the intra species relationship, how close they are, because they are same species. So it's supposed to be they will get 100 in the bootstrapping method. So what and then I construct the phylogenetic tree. Still the method and the model are UP distant. So the DNA, substitution type, I use nucleotide. You can use protein if you want. Okay, you can just translate your nucleotides DNA to the protein, but then this one I prefer DNA nucleotide. So this one I use interbranch test. Okay, and then I use neighbor joining methods. Okay, so from the software analysis that I apply, they will give you this one. Okay, they will give you the phylogenetic tree. For example, you can see here the below paha individual 1 and individual 3 very close each other. It gives you in the relationship is 99%. It's a confidence levels. Okay, if you do statistic, you know P more than 0.05. You must your analysis, your data must be in the bell curve. So these are very confidence. Okay, so the individual 1 and individual 2 very close each other. But compared to the Tanjung Malim, individual 1 and individual 3 is lesser, meaning that they have a 70%. It might be when I asked the breeder, they said these two individuals is come from different parents. I mean different mothers. Okay. So another one you can see here. Okay. You can see here Tanjung Malim 3 and Timur Tanjung Malim 3, individual 3 and Bilok Pahang, individual 3. Their intra-specific relationship was 69%. It shows that they are very far. They're not a sibling. They're not sharing the mother. They're not sharing the fathers. Okay. That maybe this happen maybe because sometimes like deer or any breeder animals, they are social animals. They like to mate each others. Sometimes you want to get a good generation, you have to mate from one species to one same species but different individual, which is give you a good DNA. Okay, so this is one of the highlight cases in Malaysia that my lab involved and apply the sequencing analysis. Just like this one, we use the DNA approach to identify, because they claim the meat was horse meat is elite to be beef, and buffalo meat is elite to be a venison. Venison actually is a deer. Okay. So to identify that, we use a sequencing analysis. Okay, and then in Malaysia, last year and this year, we have a fake halal meat scandal. It's a very big issue, and Jabatan Kimia was in the hot suit actually during that time. And then this one, after they slaughtered the breed deer, and then they packing. So they actually they packing the cow meat, but they're selling at the deer meat price. Okay, so by the sequencing analysis, it solved, alhamdulillah, it solved most of the cases that highlighted in Malaysia for the fraud cases. Okay, so thank you so much. This is my my last slide. So you can go to our social media, Kimia Malaysia, Jabatan Kimia Malaysia. Okay, we have a Facebook. Okay, if you have any question, you can just drop an emails to projkm@kimia.gov.my, and then you also can buzz us at the Twitter, our Instagram and Facebook. Okay, thank you so much. Any question?
Thank you, Dr. for your nice deliberation. If you have any question, you can ask from the audience. Actually, this your lecture was the last one, and the audience is, I think they are tired now. Thank you. Thank you very much.
Okay.
Please, Dr. Musharraf, he will conclude the session.
I'm very thankful to all speakers of today's session. Indeed, we had a very fruitful discussion as well as the presentation by the various speakers on various topics relevant to halal testing. I'm also thankful to Doc Saab because he is kindly agreed to chair the session on a very short notice. So thank all of you. Inshallah, tomorrow we will be here right on time and the tea is ready to serve. But before to end, I would like to inform you that make sure that you are going to mark the attendance properly because only those participants will get the certificate whose attendance recording properly. Okay, so that's the important thing. Okay, thank you very much once again. Thank you. So tea is ready to serve.